Okay, let's have a class. And first of all, I want to mention this. You should have the fact sheets, which is chapter 9. Okay, go back to see your fact sheet, chapter 9. I still have something here. We will have a dilution quiz, which is right here. So the idea is always chapter 10. Make sure you read chapter 10 and then finish your dilution quiz, which is take home at 10.4, page 47. This will be another work with your bench partner. So you guys find a time, work together in the library or the study room, some places, and only 2 of you submit one. But very important is you need to write down how you calculated it. It's not only the answer. Okay, you can do handwriting, you can do print, that's fine, but make sure you have a procedure how you get the answer. So the quiz is at page 47, 10.4, and the guidelines will be chapter 10. I still have some fact sheets here from chapter 9. You could get it. Okay, chapter 9, chapter 10, they're together. So if you didn't bring, please do it. Okay, you all have the urine sample results. So let's review our urine sample. Okay, so the urine sample, what we did. The first thing what we did is a dipstick test. And the dipstick test, if you still remember, we were using is a strip immersed into the urine solution. And what you are looking for is a 3 marker, is white blood cell, which is leukocyte, red blood cell, and nitrite. If one of these positive, we'll move on to a 3 agar, which is Macanchi agar, blood agar, and a neutron agar will be using for quantitatively to tell us a number. So if you look at your results, this is your Macanchi agar, what it looks like. Did you see a colony there? Yes, there's a colony. So remember how we did? We used plastic sterilized loop. We did a strip plating, and you see colony there. And on the blood agar, what do you find? You still have a colony there. And if you look carefully, you will see lots of them are a gamma hemolytic, or some of them are a beta hemolytic a little bit. So we'll see what the results, what it looks like. So this is a blood agar. Okay, the blood agar, what you see is those pinpoint colonies sometimes. And the beta hemolytic colony. Because that means you may have an invasive pathogen. Okay, the neutron agar, what we did a very gentle streak, and I said very, very gentle, and then we turn 90 degrees, we do a second streak, and then we incubate 35 degrees Celsius, 24 hours. Now obviously all of these have to be incubated 35 degrees Celsius, 24 hours. Then we look for colony. Okay, we look for these colonies. If the colony population is more than 100,000 CFU per ml, this will be clinically diagnosed as UTI. So look at your plates. Do you think it's more than 100? Oh yes, more than 100. and you can split them into two foci, and then you count one corner and multiply by 4. It's absolutely more than 100. It could be more than 300. Okay, so this case, this patient is absolutely diagnosed as UTI based on these results. And in this lab, we don't have a urine sample from the real patient. In our second lab, fourth lab, we do have some sample from our student. So we can talk in those lab sections. Okay? So that's what we have. Okay, today's lab, fact sheet is here, chapter 18, which is extracellular enzymatic activity for the bacteria. Okay, so this is what we're going to do. We want to mention several things. It's enzymatic activity of bacteria. So we talked about the energy resource for bacteria to use is mainly sugar, carbohydrates, mainly is glucose. The detail we'll talk about in the lecture. However, in the real life, they not always get glucose. They'll get lots of the other energy resource. So for example, they may get starch. Okay, what is starch? Can somebody tell me what is starch? Can you draw a picture of starch? So starch is composed by what? So H, OH, OH, H, go down, OH, H, H, CH2, OH. What is that? Glucose. Okay, so I have another glucose. I'll just draw like that. I'll be here, go like this, go here. Okay, we draw another one. Okay, then what we do? That will be H, OH. Then go here, will be H, OH. Then here, OH, H. Okay, here, H, CH2, OH. This is another glucose. Okay, what is this one called then? Carbon number 1, carbon number 4. What do we call it? 1,4-glycosidic bonds. So, what is this guy? Organic chemistry. What are these glucose combined together called?
Disaccharide.
Maltose. Yeah, disaccharides. Good. That's called a maltose. Okay, if I have a multiple maltose connected together, what is it called? Starch, is that right? And that one type of the starch, what it is? 1,4-glycosidic bonds. You will regenerate a starch is amylose. Okay, what is another type of the starch? Amylose. Of course, we could be 1,6. Is that right? That's amylopectin. So what it looks like? Can you do it? Okay, let's just do this. You just do like that. This time around, we're going to do this guy's still the same. Okay, we're not going to draw the whole thing. Okay, this guy here, we're going to go here. So we go here, here. Okay, then we come out here, CH2, OH. So we connected it with this guy. So that is called 1,6' glycosidic bond. Amylopectin. Okay, which enzyme used to hydrolyze starch? Become maltose, They further break down, become glucose. Which is the enzyme name? Amylase. Okay, bacteria could have amylase. So how we do it? How do we determine bacteria has amylase? It's very simple. We have an agar called starch agar. And we split them onto two. The left side, we will inoculate E. coli. And the right side, we will inoculate Bacillus subtilis. And after 35 degrees Celsius, 24 hours, how do you know starch is gone, starch is still there? Another organic chemistry question. How do you know? Which chemical starch will react with it? Is that right? We'll react it with iodine. Where we used the iodine before? In the gram stain. So we can use gram iodine. So that's a tricky thing. It's just going to twist your mind a little bit. After incubation, we're going to pour the iodine here. Okay, the iodine is going to be pouring here. If one side shows the brownish color, what this means, the starch is still there. If the other side is no brownish color, the starch is gone. What means that bacteria has amylase. So that's kind of like negative got positive. Is that right? So that's one of the way we're going to do. So starch agar. What's a starch agar looks like? Starch agar looks like very general. Looks like a traditional neutron agar, but you can see a little bit more white, more greenish white. We add a starch there. So that's a starch agar. So first thing, bacteria could use starch as long as there is amylase. Okay, second one. Lipids. Okay, bacteria could use lipids. What is your lipid structure? Okay, good. 1 second. Is that right? That's what this lipids looks like. CH2. Okay, CH3. That could be long and long. You learned that. Okay, that could be saturated. Okay, that could be like this guy. Oh, saturated is C18. What is that? Steric gases. It could be, go here. You may have this guy comes out. So for example, let's say C18-1. Oleic acids. And then you could have like this. Two. Oh, they'll go here too. So for example, like that. Linoleic acids or linolenic acids. Then you have a trans cis. You learned in your nutrition class, okay? We can talk this whole day. So we're not going to talk that because it's not a biochemistry class. And this number, we call it stereo specific number 1, 2, 3. Typically for some of the plant-based lipids, the middle one usually unsaturated fatty acids. Okay, so bacteria can use it. They need an enzyme. What is the enzyme? Lipase. Very similar to lipids, only change ASC called lipase. Okay, how we do it? This time we're using a very shiny, beautiful agar. That's called sprit blue agar because it's blue. So we're going to using a blue color. So what we do? Spray them onto 2. Same thing. This is called split blue agar. On the left side, we're going to inoculate which bacteria? E. coli. Right side, Staphylococcus aureus. Staphylococcus aureus on the agar plates. This is from my lab, Staphylococcus aureus. Okay? And the E. coli just on the slants, like we did a bit before. Then we're going to incubate. 35 degrees Celsius, 24 hours. It's easy, you don't need to do anything. You will see most likely a transparent zone. That means amylase, a lipase test positive. It's very simple. Because we used it, we break it down, it shows transparent. Okay, it's a very smart way to bacteria to use. So think about in the environment, if you don't have a glucose, you don't have a starch, I only have a lipids, so you have to use it. And they will grow very well. Now we're using split blue agar. There is another agar also we used before, egg yolk agar could be used. We don't have it because egg yolk is proficiency and numerously multiple different types of the lipids there. So that's why we can use egg yolk agar. Sometimes we don't have a split blue agar. Okay, that's a little bit yellow color. It's the same like we did for the you eat egg yolk. Okay, number 3. So we have starch, we have a lipase. What else we didn't have? Protein. Bacteria can use protein. Okay, so what's the protein going to be? Looks like? Well, protein is composed by amino acids, then goes to peptide, then goes to different structure. Is that right? Go to like primary, secondary, tertiary structure. We'll talk about these later on. So amino acids, we mentioned about that. Amino group, H, the difference is R, COOH, carboxy group and the amino group. So we'll mention that in the exam 3, a detail. Okay, using protein. Which food products has a lot of protein? This agar, skim milk agar. Skim milk agar. So why we use skim milk? If you go to buy a bottle of the milk, there is a whole milk. There is a reduced fat, like 5%, 2%. There is a 1% reduced fat milk and skim milk. What do you mean skim milk? Theoretically, it is 0% fatty acids. So we take out all fatty acids. Now milk, what's the sugar there? It's lactose. But bacteria won't use lactose. Okay, so skim milk agar is a good material. What we do? Split them onto 2. Left side, inoculate E. coli. Right side, inoculate Bacillus subtilis. What are you going to looking for? Same things, looking for transparent zone. If there's a transparent zone showed up, which means the milk has been broken down. By the way, what is the major protein in the milk? Does anybody know? Maybe you know. Casein. So what's the enzyme for that? Caseinase. It is one of the components of protease. Okay, so that's what we have to do. Now, what else we need to do? Don't forget label. Don't forget label, and don't forget label on the back of the agar like you did it before. Okay? Same thing as before. I will be showing you how we're gonna do this. So everybody coming to the front right here. Doesn't matter how easy it is. I will be showing you how we're gonna do this. Okay, I think it's very important when we get to show you. Okay, can somebody donate gloves for me? Yeah, if somebody have a gloves. Thank you very much. So again, first thing first, clean your bench, wear gloves, and go ahead to get you 3 plates. And when you see these agar plates, we pull out at the refrigerator. So the first thing first is always don't forget to clean them up. So we clean them at the bottom, and I say that the condenser, you have to remove it. It's a good thing is that this one is only for yes or no, which means there's a transparent zone or no transparent zone. It doesn't need to be really we see the population, which is it's a qualitative test, not a quantitative test. Okay, so first thing first to clean it. It's very wet. Okay, when you have this, throw away in the trash can. So we need somebody have a Sharpie pen. Do you have a Sharpie pen? Just okay. Skin milk agar. Split them onto 2. Split blue agar. Split them onto 2. Starch agar. Split them onto 2. So write your name, initial, date. So write your bench number, initial, date. That's very important. The first information. What are you going to put the bacteria there? E. coli, Bacillus subtilis. E. coli. For this guy is what? E. coli and Staphylococcus aureus, so SA. The last one, E. coli and Bacillus subtilis. Okay, that's a label, very simple. Now, you're going to make sure it is in the same corner. Okay, so you can see it. And this side is E. coli, this side is the other bacteria. So now what you do is using your Bunsen burner. Thank you. I'm going to turn it on. That's good. We're going to go back to use loop. Okay. Make sure it is sterilized. It is sterilized. Okay, we're going to do E. coli first. Let's do E. coli. Flame the loop. Gentle pick bacteria. Okay. Put on the side. Which side is E. coli? This side is E. coli. So very gentle. That's all. Okay? E. coli is right here. Flame the loop. Get the bacteria. Which side is E. coli? This side is E. coli, so very gentle. That's E. coli. Okay, flame the loop again. Get the E. coli. So where you go? Let me show you one more time. Okay, this one I have to do like this. Okay, just very gentle streak. That's all. That's E. coli is done. Okay, put on the side. Second one, bacillus. What are the two for bacillus? Starch agar and the milk agar. Okay, bacillus. Go down, get the bacillus. And this side, so we do streak right here. Okay, blend the loop. Sterilize. Touch bacteria. And then in here, we're going to do it. That's all. Very gentle. Okay, which one we haven't done? Is Staphylococcus aureus. It's going to be on the plates. So what you do? Hold down, pick colony. Okay, pick up a colony, don't damage it. Then it is very blue one. That's why I put the blue in the middle. Very gentle we do this. That's all. And then give it to TA. We'll do the incubation for you. And we'll see it next week. I hope by the time you see a very transparent zone, but no contamination. If you have a cross contamination, next week Tuesday looks horrible. It will be everywhere.